Neuronal cell migration. Since all cortical neurons originate near the ventricular surface of the cerebral vesicle, they must all move to their final positions in the cortex, which develops in the outer regions of the cerebral wall, just below the pia. After their last division, postmitotic cells become polarized, extending the leading processes toward the pia and then translocating the nucleus and surrounding cytoplasm within that process. The extension of the leading process and nuclear translocation are inseparable cellular events. Initially, while the cerebral wall is relatively thin, the tip of the leading process can reach the cortical plate, and the nucleus needs to move only a short distance in both the small rodent cerebrum as well as in the human at comparably early embryonic stages. However, during the subsequent course of corticogenesis, the cerebral hemispheres enlarge and the length of the migratory pathway increases, particularly in the large primate cerebrum, in which, during mid-gestation, a massive migration of neurons occurs concomitantly with the rapid growth of the cerebral wall. This magnitude of cell movement is perhaps the reason that neuronal cell migration was first observed in human embryos. His 1874. When this large increase in length occurs, bipolar migrating neurons do not span the entire width of the cerebral wall. Subsequent time-lapse imaging studies in the mouse forebrain have shown that this way of migration, termed somal translocation, occurs in all cells irrespective of the length of the migratory pathway. In the early 1970s, it was discovered that postmitotic neurons find their way to the cortex by following elongated shafts of radial glial cells, figure 1.3. Radial glial cells are particularly prominent in primates, including human, whose fibers span all the length of the convoluted cerebral hemispheres of the late stages of corticogenesis. While moving along the glial surface, migrating neurons remain preferentially attached to curvilinear glial fibers, a finding which suggested a gliophilic mode of migration that may be mediated by heterotypic adhesion molecules. As many as 30 migrating GFAP negative neurons have been observed migrating along a single GFAP positive radial glial fascicle in the human forebrain during mid-gestation. However, some postmitotic cells do not obey glial constraints and move along tangentially oriented axonal fascicles. For example, black horizontally oriented cells align with thalamic radiation, TR in figure 1.3. We suggested the term neurophilic to characterize the mode of migration of this cell class. So the other one was gliophilic. Although lateral dispersion of postmitotic neurons was initially observed in Golgi stain preparations, for example, figure 1A of the report of the Boulder Committee, so that was figure 1.1, it attracted renewed attention after the characterization of the migration through the rostral migratory stream from the postnatal ventricular zone to the olfactory bulb, and from the ganglionic eminence to the dorsal neocortex. Studies in rodents also suggested a more widespread dispersion of clonally related cortical cells. Although in rodents, most of tangentially migrating cells in the dorsal neocortex are inhibitory GABAergic interneurons, there are also a number of migrating oligodendrocytes. It should be underscored, however, that clonal analysis of the convoluted primate cortex revealed that the majority of migrating cells, both projection cells and interneurons, obey the radial constraints imposed by the radial glial scaffolding. And the following section on the radial unit hypothesis. Oh, whoops. Ignore that one. Also, lineage analysis of transcription factors specific for either dorsal or ventral neocortex progenitor cells suggested that the majority of interneurons in the human cortex are derived from ventricular zone, subventricular zone of the dorsal neocortex. This finding strongly suggests that the primate cortex may confer species-specific differences in basic developmental events compared to other mammalian species. Considerable progress has been made in understanding the molecular mechanisms behind neuronal migration and the physical displacement of the cell perichera during somal translocation across the densely packed tissue. Initially, based on an observation in situ, It was proposed that a single pair of binding complementary molecules with gliophilic properties can account for recognition of glial guides. However, in recent decades, many classes of recognition and adhesion molecules have been discovered and are being tested. The subject is too large to be reviewed in detail here. However, it is important to mention that voltage and ligand-gated ion channels on the leading process and cell soma of migrating neurons regulate the influx of calcium ions into migrating neurons. Calcium fluctuations in turn may trigger polymerization of cytoskeletal and contractile proteins essential for cell motility and translocation of the nucleus and surrounding cytoplasm. It was clear that translocation of nucleus requires cytoskeletal rearrangement, and a host of molecules involved in this complex process have been identified. For example, double cortin and LIS1 have been shown to be involved in cytoskeleton dynamics during neuronal migration, and their mutation in humans has been implicated in certain brain abnormalities such as double cortex and lissencephaly type 1. Together, these studies indicate that neuronal migration is a multifaceted developmental event involving cell-to-cell recognition, differential adhesion, transmembrane signaling, and intracytoplasmic structural changes. Nuclear movement during both radial and tangential migration is a saltatory two-step process alternating between resting and dynamic phases. This process is evolutionarily conserved and involves a microtubule organizing center called a centrosome that controls microtubule polymerization. In radially migrating neurons, the centrosome controls the formation of a microtubule network that surrounds the nucleus, the so-called perinuclear cage, and establishes a physical link between centrioles and nuclear membrane. A similar mechanism has been described in cells of the subventricular zone that migrate tangentially to the olfactory bulb in the postnatal brain, and more recently in radially migrating cortical neurons. Together, these results suggest that actin myosin cytoskeleton might control nuclear movements in neurons exhibiting multiple radial and tangential modes of migration. A simple model of selective molecular components involved in cell migration is provided on the diagram in Figure 1.4. The initial discovery of the glial-guided radial migration in primates led to the proposal of the radial unit hypothesis, which has served as a useful working model for research on the cellular and molecular mechanisms involved in normal and abnormal cortical development and evolution, and will be summarized in the next section.